Hello, people.
Hello.
Can we have everyone bring your faces up here? Okay. Who will I tease today? I'm not going to tease anybody today. Oh, I'm just glad you're all here. I'm glad we could share this time. I'm glad I'm getting paid for it too. Okay, so 11:02. We only have 26 people here, which is unfortunate for the people who haven't come because I've got a quiz for you today for which you can earn one bonus point. One bonus point. This is something new I'm going to institute. Every day we'll have a quiz and you can earn a bonus point by doing it, and that's going to make people want to do it. So I will show you where the quiz is. Where are we? Hang on one second. This is my Moodle page. Editing off. Okay. So when you get here, go to Moodle, and here it is. Practice quiz 3/23/21. So I'm going to give you until 10 after to do that, which gives you 7 minutes. So go ahead and do that. And then afterward, we'll talk about the answers, and then we'll have our lecture for today. So if you've shown up late, there's a quiz in Moodle under the quiz section, and you've got until 10 minutes after to do it.
Dr. Slish, where is it? I don't see it.
Let me back up.
What?
I wasn't talking to you.
I heard the F word just now. Did I just hear the F word?
No.
Okay, quizzes. It's under quizzes in Moodle.
Yeah.
Practice
Oh.
quiz 323.
Okay.
I'm okay with the F word, by the way.
No, I was just telling my husband to be quiet.
Okay. Okay, anybody need more time?
Yeah, like another minute.
Okay, another minute. Okay, people, time to submit. Submit. That quiz down. And we can talk about the answers. We got 30 attempts. Did everyone submit? Who has submitted? Who has not submitted? Submit to me. I love that word. Finished, 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 in progress. Kozlowski, finish. Benatti, finish.
I went to submit it, but then you hit it and then it said it was hidden and wouldn't, it just
Oh.
didn't.
Damn it, I'm sorry. I'm sorry. Let me go back and unhide it and then see if you can
Yeah,
see.
that happened to me too.
Damn it, damn it. All right, can you submit now? Or did you lose all your answers?
I can submit now. It saved everything.
Okay,
Uh, I think I lost everything.
So did I.
who just said that? Who's just said so did I?
Christy,
Rachel.
Can you say it again?
Rachel.
Rachel, listen to that. Say it again. Talk to us.
It was me.
Is everybody else getting that reverb? That sounds really cool. All right, what the hell do we got here? Okay, so a whole bunch of you are in progress. All right, I will. We'll save your names and I'll give you credit for it, even though you didn't get a chance to submit it. Oops. Rachel,
Was that-
can you talk a little more? Just give us something to do. Rachel, Corley, sorry.
What, Slish?
Where are you getting that reverb from? That is so cool.
We're in the same room, that's why.
Who's in the room with you?
Anthony.
Anthony. You and Anthony together, okay. And that's giving you that reverb. That's crazy. Okay, so I've saved all the people who were in progress. So I will give you credit for it, even though you didn't get to submit. So let's look at the answers now. Okay, when a receptor is overstimulated, it becomes downregulated. When this occurs, the blank subunit of the G protein, that would be the beta gamma subunit, recruits the blank, that'd be the beta adrenergic kinase, to the membrane, which phosphorylates. Mine are all in the order of the answers here. Did everybody else have it that easy, or was it just mine? All right, don't talk to me. I'm mad at
Slish,
you.
did you mix up arrestin and phosphorylates on there?
Yeah, he did.
Which, no. I'm the professor. I know what I'm doing. Match the term with this definition. Removing receptors from the membrane is downregulation. Putting receptors into the membrane is upregulation. Excessive drug effects due to overexpression of receptors is supersensitivity. Loss of drug effect due to loss of membrane, that would be desensitization. Loss of effects over time, tachyphylaxis. So this is one that, I'm not sure I talked about this in class. I was kind of hoping that the two things. Number one, the words would kind of help you get them in the right place just because it makes sense that way. But also that if you didn't know the answers, you would learn it through this process of doing this. The one word especially I didn't talk about was tachyphylaxis. Oh, excuse me. So the thing about this whole receptor regulation thing is in your body, normally it doesn't happen that much. I can give you examples of when it does, but typically we use this when we're talking about drugs because if you give a drug that's an agonist, it will cause downregulation, and that is desensitizing the tissue. So they're kind of two different things there. If you give an antagonist, it blocks the receptor. So the receptor is not active, it doesn't get downregulated. The cell's getting less stimulation, so that causes an upregulation of receptors. So you get more receptors. Now, if you take that antagonist away, you've got more receptors there. That is supersensitivity, because now you've got more receptors and the tissue is more sensitive to the hormone. And then tachyphylaxis is like the general term that covers both of these. So whether it's an agonist or an antagonist, the more you use that drug, the less effect it has because of receptor regulation. So tachyphylaxis is both desensitization and supersensitivity, depending on, it could be either depending on what drug you're talking about, whether it's an agonist or an antagonist. Okay, match the hormone type to its release definition. Hormone released from one cell onto another cell. That would be paracrine. Into the bloodstream is endocrine. I think this one's pretty easy. Releasing and responding it to itself, autocrine. And that leaves only juxtacrine. Let's see what I got. I'm sure. Final answer. Woohoo! I got them all right. I'm going to pass this class. Okay, so you needed to get 3 out of 3 to get that bonus point. So I'll check that later, see how everyone did. And again, I'm going to do this at the beginning of each class from now on, both to give you some practice and also to encourage you to come to class. So this is what we're going to do. All right, so let's go ahead and learn something today. Let's get the learning started. Let's kick it off. Oops, wrong PowerPoint.
Did you say you have to get 3 out of 3 to get the points?
It's a high bar. It's a high bar. Nothing's free in this world. Okay, here we go. Okay. So we finished up last time talking about the different sites on the receptors and what they do. You've got the ligand binding site, which is out here. The sites that activate the G protein, which are here. And then the phosphorylation sites for receptor regulation in the tail. So I don't even know if this is right anymore. It could be. I don't know. The book changes on a regular basis. I don't know if that's right. But anyway, this is what these receptors look like. Blah, 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 blah. And now we're going to talk about G proteins. So these receptors are called G protein coupled receptors because they all interact with a G protein. And we'll talk about what G proteins are now. But basically, they're an intermediary protein between the receptor and the effector, and the effector is the thing that's causing the change in the cell. I'm just pulling up my notes here so I don't get lost and not tell you something. Okay, so G protein coupled receptors are everywhere in your body. Every single cell has got several different types of this. They're very, very common. Some are used for physiological changes in your cell. Actually, that's what most of them are. And that can be coupled to endocrine hormones or paracrine hormones or even locally released hormones. So they're very common. One kind of receptor you wouldn't think of as a receptor, at least in terms of a hormone receptor type receptor, are the ones in your nose and your mouth. So your smell receptors are all G protein coupled receptors. And actually, I hate to be a name dropper kind of a guy, but I worked with a woman at Harvard who got the Nobel Prize for the olfactory receptors in the nose. Can't remember her name now. But anyway, in that case, the ligand would be some kind of odorant in the air that binds to the receptor, and the receptor interacts with the G protein. So when you think about smells, one of the things that always comes to mind is the fact that you get used to smells really quick, right? I mean, I came into this, I'm in the bedroom here. I came into the bedroom. I'm like, that dog stinks. He needs a bath. I've been sitting here for 10 minutes. I don't smell him anymore, you know? So they downregulate or desensitize really quickly your smell receptors. But anyway, yeah, they're everywhere in your body. Mostly what I talk about here are ones linked to cyclic AMP and then IP3 calcium, because those are the two that are involved in your sympathetic and parasympathetic nervous system. Sympathetic tends to activate the production of cyclic AMP. Parasympathetic is IP3 and calcium. And those two second messenger systems are typically opposed to each other. We call that physiological antagonism. I'll talk about that later. But if cyclic AMP causes relaxation of the smooth muscle, IP3 calcium causes contraction. So sympathetic and parasympathetic systems are physiologically antagonistic because their second messengers are physiologically antagonistic. They do the opposite things in the cell. Okay, so G protein coupled receptors. The thing that makes all G protein coupled receptors the same is that they link to G proteins, obviously. So what is a G protein? We've seen G proteins already in chapter 13, RACRO and CDC42. Those are G proteins. And I think we talked about G protein cycling there too. Those, and also we'll see RAS later in this chapter, are small monomeric G proteins. So they're GTP binding proteins, but they're monomeric. The ones we're talking about now are large heterotrimeric G proteins. So heterotrimeric, there are three different subunits to it, and each of them is different from each other, hetero. You've got the alpha subunit, and alpha subunit is kind of the business part. It does most of everything that goes on with these G proteins. And then you've got the beta and the gamma subunit. The beta gamma are always attached to each other, so they don't separate. So you've got the alpha subunit and the beta gamma subunit. The alpha subunit, I call it the business part because it's the part that actually has the G protein binding, the GTP binding site. So the alpha subunit binds either GTP or GDP or GTP, right? One or the other. When it's got GDP bound, it's inactive and it binds to the beta gamma subunit, and that kind of shuts the whole thing down. When it's got GTP attached, then it separates from the beta gamma subunit and it goes off to produce effects in the cell. Typically, it's going to bind to some enzyme and turn that enzyme on. So that's your alpha subunit. The beta subunit then is kind of like, we always used to say it was kind of regulatory. It just kind of holds on to the alpha when it's inactive. There are some roles for the beta gamma. So when the alpha and the beta gamma separate, sometimes this will go off and activate something. For example, we saw in the last class that when a receptor is active, it'll activate the G protein. The G protein then separates alpha from beta gamma, and the beta gamma then activates that BARC, that G protein coupled receptor kinase. Let's back up and look at that real quick. So here, that's right, I didn't have the animation for this, but when the alpha separates from the beta gamma, let's just do this. When the alpha subunit here separates from the beta gamma, it's the beta gamma that brings this GRK up and causes phosphorylation of it. So that's one role for the beta gamma. Also, the beta gamma can activate some potassium channels, and that causes effects. But for the most part, we think of the beta gamma as the regulatory part, but it does have some roles too. Okay, so how does this cycle work? When the cell is inactive, when the receptors are inactive, all the G protein will be in this state, or most of it will. I mean, there'll always be a little bit of baseline activation of this thing, but for the most part, it's inactive. When the receptor gets activated, it becomes a GNRP or a GEF. I talked about this back in chapter 13. Guanine nucleotide release protein, or guanine nucleotide exchange factor. Okay, one or the other, it doesn't matter. What you need to know is that this is an activated GPCR. So that little activation domain kind of sticks out and it jabs into the alpha subunit, which opens it up, and the GDP goes flying off. Once the GDP comes off, the GTP comes on, and then it separates out and then activates something. But that will only last for a little while. In the small monomerics, I said there's this other protein, GTPase activating protein, which can stimulate the GTPase activity in this alpha subunit and have it break it down into GDP. In these receptors, it's not as important. That gap protein may or may not be there depending on the cell. For the most part, in these large heterotrimeric G proteins, you ought to think that the alpha subunit has got this inherent GTPase, and it will hydrolyze GTP, phosphate comes off, and it goes back into GDP. And then that shuts it off. So that's in general how it works. Now, steady state, what's going on in the cell is when you've got hormone around, you've got a bunch of activated receptors. And so each of those receptors will then activate a bunch of G proteins. And these G proteins constantly, once they get turned on, will constantly turn themselves off. As long as you've got an active receptor there, it will cycle through and get turned on again. So it's constantly turning off and turning on. Okay, so it's going through the cycle as long as there's hormone on the receptor. When the hormone goes away, it gets stuck in an inactive state and it stays there. So that's your G protein activity. It's between the receptor and the effector, and it's linking those two together with this hydrolysis being the mechanism of shutting itself off. Okay, and here we are doing that. Let's do it. Okay, so here's your generic receptor. Here's your G protein with GDP attached. Ligand comes on receptor. This then becomes a GEF, GEF, interacts with the alpha subunit, kicks GDP off. The alpha subunit then picks up GTP, and it goes off and activates some protein. And your beta gammas there is being it's been being kind of sequestered by this protein, but it's just hanging out there. While it's on this protein activating it, it will hydrolyze its GDP into GTP and come back and rebind to the beta gamma. And then cycle through this over and over again. As long as you've got ligand on receptor, when there's no more ligand, it stops in that spot and it's ready to go next time. So that's the general thing that, that's the general G protein coupled receptor activity. Any questions about that? Comments? Discussion points? No? Okay. Well, vamos a continuar. Let's go, people. Vamos. Vámonos. Whatever. Okay, so the next section that I'm going to talk about different second messengers that are linked to different second messengers that are linked to their receptor through a G protein. And like I said earlier, the two main ones I want to talk about are adenyl cyclase and IP3 calcium. So here is adenyl cyclase. It's the enzyme here, I always say AC for adenyl cyclase. AC will take ATP, here's our ATP, and turn into cyclic AMP. I kind of want you to know these structures because I think it's important. So ATP then, if you know from biochemistry, these little squiggle lines mean high energy bond, right? So you've got high energy bond here, high energy bond here. Why is it high energy? Look at all these negative charges you shoved into the same spot, right? You've got 1, 2, 3, 4 negative charges all put together. Takes a lot of energy to get them there. So ATP is really high in energy. So adenyl cyclase now is going to take this phosphate and take this oxygen and do a nucleophilic substitution here and bring this OH off. And these guys will be a leaving group over here. So that PPI or pyrophosphate comes off, and then you've got now a circle between the 3 and 5 carbon of the ribose of that nucleotide, okay? Nice 6-member ring there. So that is a phosphodiester, right? An ester is a double bond O to C, double bond OC. We've got 2 of them here, phosphodiester. Pyrophosphate now, this section over here, each of these squiggles is worth 7.5 kilocalories per mole. This is broken down during the reaction, and then this is acted on by pyrophosphatase that'll break this bond. And so that's the energy that drives the reaction forward, using up all the energy of 1 ATP. Okay, here's our cyclic AMP. By the way, let me mention something here before I forget it. You all need to know, the most important thing for you to know in this class is to not do the things that annoy Dr. Slish. So here's what bothers me about cyclic AMP. Do not at any time write this and call it cyclic AMP. It is not. This is camp. Camp is where you go to like shoot the bow and arrow and do the canoe and, you know, sing songs around the campfire. Okay, that's camp. Do not write that. Cyclic AMP is small c AMP, okay? You're not going to fail college if you do it wrong, but you're going to annoy me, and that's probably just as bad as failing college. Okay, so here's cyclic AMP, and now to break this down, right? Every car has got both a gas pedal and a brake, right? So you need a brake. Once you've activated cyclic AMP, cell responds to it, and then you need to break it down so the cell will stop responding when the hormone's gone away. How do we get rid of the cyclic AMP? This is a phosphodiester, so our enzyme is phosphodiesterase, okay? Easy as that. So phosphodiesterase is going to break this bond, adding water across it, and then you're back to good old AMP, right? So AMP then will be acted on by other enzymes and regenerate ATP from it. So that's adenyl cyclase. ATP, adenyl cyclase then makes that circle, cyclic AMP, and then phosphodiesterase breaks it apart. So you're back to good old AMP. All right, so how do we make this part of a receptor system? So here's our receptor. And I usually use the example of epinephrine activating a beta adrenergic receptor. Because I'm a cardiovascular pharmacologist, and that was really important, still is really important. So it's just a really good example. But there are a lot of different hormones that will activate their own receptors, and those receptors will be linked to cyclic AMP as well. So here's our beta adrenergic receptor. The ligand, epinephrine, binds to the ligand's binding site. That activates the activation site. That then causes the alpha subunit to activate. It then binds to adenyl cyclase. Adenyl cyclase makes cyclic AMP. And this is just the G protein cycling. So what does cyclic AMP do then? Once cyclic AMP is produced, it's going to activate the enzyme protein kinase A. Protein kinase A is this bad boy. So protein kinase A is a serine threonine kinase. And that distinction is kind of important. So most kinases are going to be serine threonine kinases, which means they'll phosphorylate proteins on serines and threonines. This one in particular will phosphorylate proteins in sites like this, KRX, S, or T, or RRX, S, or T. Okay, so what this enzyme is going to do is go out and look for proteins that have this sequence in them. Lysine, arginine, anything, and then a serine or a threonine. Okay, so you can imagine these positively charged amino acids are binding somewhere near the active site of the catalytic domain. And then right next to that, it's got an ATP and it's phosphorylating the serine or threonine that's next to it. Okay, or, you know, two positive charges, RR, and then a serine or threonine. So when there's cyclic AMP in the cell, it comes out and binds to regulatory subunits of the protein kinase A. And those regulatory subunits then will become detached from the catalytic subunits. And these two are detached from each other. So these two go off and they phosphorylate any protein that's got these sequences on them. So that's cyclic AMP. So let's go over that again, because I went over it kind of fast. When I do it in the class, this takes a long time because I draw it all out and I add more information as I draw it. I have all that information in your notes that are posted there, so you can read through that. Let me bring it up and make sure I don't miss anything. Okay, so your hormone binds to its receptor and that activates it. The active receptor then activates the G protein, interacting with the receptor. The alpha subunit then diffuses across the membrane, right? It's attached to the membrane with this lipid, so it stays with the membrane, comes over to adenyl cyclase. Adenyl cyclase then makes a bunch of cyclic AMP. Cyclic AMP will then activate, each cyclic AMP molecule basically activates one protein kinase A, and then the protein kinase A goes off and phosphorylates a whole bunch of proteins. And that causes the changes in the cell. So what I really want you to know about this, besides that pathway from here to here to here to here, that's the easy part. The more in-depth understanding of this is, again, the steady state of what's going on. So it's not like a hormone binds and 1-to-1-to-1-to-1 response. Boom. No, it's always like a whole lot of things going on at once. So let's talk about the steady state of it. So, and this is important in terms of amplification of the signal too, versus things that aren't amplifying. So in your notes, I talk about stoichiometric versus amplification. So stoichiometric is when you have one molecule binding to one molecule and activating it, okay? That's 1-to-1. You're not amplifying the signal, it's just 1-to-1. Amplification is like, now this protein is active and makes a whole lot of something else, right? That's amplifying the signal. So what I need you to follow here is which of the steps in the activation of this pathway are stoichiometric and which ones are amplifying. So starting at the beginning, you've got your epinephrine that binds to the epinephrine receptor. Okay, that's stoichiometric. Okay, 1-to-1. And that activates the receptor. Now the receptor, once it's activated, will activate every G protein that bumps into it. So one receptor may activate 100 G proteins. Okay, that's amplification. Okay, so now we've gone from one molecule to 100 molecules active. Now that alpha subunit, once it's got GTP bound, is going to slide across and bind to one adenyl cyclase. That's 1-to-1, that's stoichiometric. Okay, once you activate adenyl cyclase, it's going to convert 100 ATPs into cyclic AMP, okay, before this thing turns itself off and goes away. That's another amplification step, okay? So when this thing shuts itself off, and the third step here, the third part of this is cyclic AMP then will bind to the regulatory site of protein kinase A. That's stoichiometric, right? 2 cyclic AMPs, 2 regulatory subunits, 2 to 2 is like 1 to 1. So it's basically just a 1 to 1 interaction. So that's stoichiometric. That releases the catalytic domains, and these catalytic domains will go off and phosphorylate 100 proteins apiece or something. You know, I'm kind of making that number up, but that's another amplification step. So when you're looking at these mechanisms, you probably want to draw them out yourself so you can see it yourself on paper, and look at each step and see which step is catalytic and which steps are catalytic and which are stoichiometric. Another thing you need to know about these mechanisms is how does each step shut itself off? You know, the way this process works is the hormone hits the receptor and it produces a response. Eventually that hormone is going to go away, and then the response has to go away too. Otherwise, you know, you turn something on, it just stays on forever. So how does each step turn itself off? Well, the first step is the ligand binding to the receptor. And this is always non-covalent, okay, which means the hormone binds to the receptor. And like we said last time, you've got functional groups in the hormone binding to functional groups in the side chains of the receptor. And it's usually, there can be salt bridges, but mostly it's going to be hydrogen bonds, which are somewhat strong, but they're not covalent, nowhere near covalent. So molecules are always moving, right? As long as you're above 0 K, molecules are always moving. Eventually this thing's going to pop off. So it's not going to stay on forever. It'll bind to it and eventually come off. Now, if you have a concentration of hormone outside the cell, another one's going to jump on, right? So that's kind of the receptor binding thing. It's like you've got receptors there and hormones will bind to it, jump off, bind to it, jump off, bind to it, jump off. So it's constantly being activated like that. And it inactivates when it comes off. And in the body, your hormones release, but it'll eventually be taken away. And when that last hormone molecule comes off their last receptor, now all the receptors are off. Okay, so reversing this is basically the ligand jumping back off the receptor and the extracellular medium being cleared of ligand. So now this receptor stays off. Now, when the receptor was on, we had this G protein cycling happening, right? When the receptor was off, then this system gets turned off by this GTPase activity, which turns it back into beta, into the inactive form. Okay, so that's how that step gets turned off. And when that gets turned off, when this goes to GDP, it comes off the adenyl cyclase, and that turns adenyl cyclase off. It stops making cyclic AMP. How do we get rid of cyclic AMP? Phosphodiesterase is there. So the way you want to think about phosphodiesterase is it is always there and it is always active at pretty much the same level. So when you stimulate the production of cyclic AMP, it goes up, but it only stays up as long as you keep making it, because it's constantly being broken down at a lower rate than what is being made, so it's higher, right? Well, pretty much actually when it peaks, then the production and the destruction are at the same rate, so it's staying at a high rate. When that G protein comes off adenyl cyclase and it turns off, the concentration of cyclic AMP drops in the cell, because now you're not making it anymore, you're just destroying it. So the way we shut that step off then is because phosphodiesterase is going to turn it off. Okay, then the last step, how do we turn this off? Well, just like the ligand bound to the receptor, cyclic AMP bound to this regulatory subunit, it's going to jump on and off. As long as you've got cyclic AMP inside the cell, whenever one jumps off, another one will jump back on, right? When cyclic AMP drops in the cell, all the cyclic AMPs come off the regulatory subunits, they get broken down, and then the catalytic subunits bind to the regulatory subunits, and the kinase shuts off. One more step. So the phosphorylated proteins then, the catalytic subunit went off and phosphorylated a whole bunch of proteins, right? Well, another thing like phosphodiesterase is the phosphatases. Let me write that down. So phosphatases. So the phosphatase is the opposite of the kinase. So the kinase is taking ATP and putting phosphate on a protein, and the phosphatase is going to take the phosphate off. So if the kinase turns the thing on, the phosphatase turns it off, or vice versa. Phosphatases are kind of like that phosphodiesterase. They're always in the cell. They're always active at the same level. So if you want something to happen in the cell, you have to keep phosphorylating the important proteins. When you stop phosphorylating those proteins, the phosphate will be taken, well, the phosphate is constantly being taken off the proteins, but once you stop rephosphorylating, they all get dephosphorylated and they go back into inactive. So that's kind of the steady state of it. When the hormone's on the receptor, you're constantly making ATP or cyclic AMP and making it and breaking it down, constantly activating phosphorylation. When the hormone's gone, cyclic AMP drops and the phosphorylation state drops. Okay. Any questions about that? I made that a lot more complicated than it needed to be, I think, but I think it's more fun that way. Okay. Anyway. All right, what does this do in the body? This is cell biology. You don't have to know what goes on in the body, but it's kind of interesting to know, what's the connection. So in the liver and muscle, epinephrine is kind of doing the opposite thing that that insulin does. So insulin makes you take glucose up and turn it into glycogen, right? Epinephrine is going to do the opposite. It's going to make you break down glycogen and transport it out of the cell. So epinephrine, think about it as your fight or flight. You're in trouble. You need to get out of trouble. Get some glucose in your blood, right? Get the glucose up so we can have more energy and do more running. Because I know you people, you're more runners than fighters. I just sense that. You're college people. Okay. What else? In the adipose tissue, epinephrine will cause fatty acids to, triglycerides to break down to release fatty acids. Again, energy production. Cardiovascular system is going to increase your heart rate, increase your blood pressure, relax your bronchial tubes, get more oxygen, more blood flowing, get more oxygen out to your tissue so there's more energy. In the kidney, you're going to reabsorb water. But this is a different hormone. This is anti-diuretic hormone. I talk about this in a lot of classes, and every time I bring it up, it's like, did I talk about this yet? So ADH is released by your brain when your blood gets too hypertonic, right? So you release anti-diuretic hormone, you put those aquaporins into the collecting ducts, and you bring water back into the body. So that's where I talked about it back in the chapter 8 with aquaporins. So the ADH hormone then causes those to go into the membrane and you reabsorb water. Parathyroid hormone. I discussed this a little bit when I was talking about calcium metabolism. If you look up calcium metabolism, you'll find parathyroid hormone, because calcium metabolism typically means on a physiological level in your whole body what's going on with calcium. So if the calcium levels in your blood drop, pretty dangerous thing, calcium's really important. Your brain will release parathyroid hormone, which will cause breakdown of calcium phosphate, and calcium comes into your body. So your bones are used as a buffer system for calcium. And because, if your calcium levels drop, your heart stops beating, your muscles stop moving. So calcium is really important to control. Okay, question. Would an increase in epinephrine cause an increase in fat burning in the body? I believe so. Because it says here it will cause breakdown of fatty acids, and those fatty acids are going to be used, I imagine. So I think so. Although, I don't think I would use that as a weight loss system. Epinephrine will drive you nuts. Okay, that's where you're going. Sorry. Okay, so that is cyclic AMP. Any questions about cyclic AMP and that whole schmear. No. Okay, so the next hormone system is something we've talked about obliquely. I love the word obliquely. We've talked about obliquely over and over again in this class so far. And it has to do with that calcium metabolism, cellular metabolism of calcium that I talked about over and over again. So this is all centered on this phospholipid that, again, I keep bringing it up because it's so goddamn important, which is PIP2. So PI is phosphatidylinositol. So that's one of the lipids that's made in the smooth ER, right? And that would be this structure here, okay? So let me just, I'd like you to know this structure too. This is pretty important. So phospholipids in general are a glycerol molecule with 2 fatty acids on it, right? And this one here is usually arachidonic acid, which is 20 carbons long. So glycerol with two acyl groups, basically fatty acid acyl groups. And then it's got a phosphodiester bond to some polar group, serine, ethanolamine, choline, and here inositol. Inositol is a 6-member carbon ring, cyclohexane, with an OH on each of the carbons, which means this is a sugar now. It's no longer cyclohexane. So you've got this 6-member carbon ring with an OH on each carbon. So we number these carbons 1, 2, 3, 4, 5, 6, like that. 1, 2, 3, 4, 5, 6. So that represents probably 5% of your membrane is PI. Some of that will be phosphorylated into, PIP. If you put 1 phosphate on this inositol, let's see if we can block this out. Again, you're seeing behind this filter here. Dr. Slish works its magic. Okay. So you put 1 phosphate on that, and now you've got PIP, okay? Phosphatidylinositol phosphate. And that's usually in the 4 position. And then you can phosphorylate it again. Now you've got PIP2. So PIP2 represents maybe 1% or less of the phospholipids in the membrane. So it's kind of rare, but look at this son of a gun here. It's got, this is basically 1.5 negative charges, okay? Because based on the pH inside the cell, one will be phosphorylated. One will be protonated half the time. The other one will never be protonated. So this is like 1.5 negative charges here. So is this, and this is 1 negative charge. Look at all these negative charges in one place. That's pretty attractive, right? Any kind of protein with a lot of positive charges is going to suck right up to that. And that's what we talked about in chapter 14 when we were talking about a regulation of actin. And our enzyme at that time, our important enzyme was PIK, phosphatidylinositol kinase. So PIK puts those phosphates on there. And there are several different isoforms of PIK. There's not just one, there's several different isoforms and different ways to activate it. We'll see PIK later in this chapter. Okay, so PIK puts those phosphates there, and now you've got PIP2. Now this thing can be divided here by phospholipase C. Phospholipase C will break this bond and release inositol with 3 phosphates, or IP3, okay? So IP3, again, is gonna link to something we talked about, the IP3 receptor on the smooth endoplasmic reticulum. So the IP3 receptor is similar to the ryanodine receptor. It's a huge channel in the smooth endoplasmic reticulum. And when IP3 binds to it, it opens and calcium comes flooding out of the SR or ER, smooth ER. So that's our PIP2 and going into IP3. Now, another thing that's going to happen here is when you cut that bond you end up with this left over too, right? So IP3 comes off, but also glycerol with two big acyl groups on it, diacylglycerol. So this is also going to be a second messenger. So this is a twofer. Okay, you activate phospholipase C and you get two different second messengers, IP3 and diacylglycerol. DAG is going to go off and activate a kinase, protein kinase C. So we saw protein kinase A, this is protein kinase C, we'll see protein kinase B later. So DAG will activate protein kinase C and it will go off and phosphorylate some things and activate different things in the cell. Okay, so this is PIP3 or PIP2, phosphatyl and inositol bisphosphate and phosphorylated on the 4 and 5 position. Phospholipase C breaks that off and you get these two second messengers. So let's see how this works in the real world. Okay, again, very similar to the cyclic AMP system, except now let's throw a different receptor here. Let's make this an alpha adrenergic receptor. So we'll talk about norepinephrine as the ligand. Norepinephrine is the endogenous neurotransmitter of your sympathetic system, norepinephrine. And norepinephrine will bind to the alpha adrenergic receptor and activate a different G protein. Okay, so let me back up a tad because I forgot to say something up here. Here. So with cyclic AMP, this G protein is one subtype of G protein. So this is called GS. Okay, GS. G sub S. So this G protein is called GS because it's stimulatory. It activates cyclic AMP. This bad boy here, oh, let me do that. Hang on a second. You're so lucky, people. You get to glimpse behind the curtain here and see how Dr. Slish works his magic. Oops. This is going to be a different G protein. Come on, you. Damn you. Damn your eyes. This is GQ. Okay, so this is an isoform of G protein that's going to activate phospholipase C. So this alpha receptor will only activate GQ, it won't activate GS. And the beta receptor only activates GS. So, and there are several other receptors that will also activate GQ or GS. There are several different receptors that will go through the same G protein and activate the same second messenger. But just looking at this one, we've got norepinephrine activating the alpha receptor, which is then activating GQ. GQ then will release GDP, pick up GTP, come over and activate this bad boy, which is phospholipase C. Phospholipase C then breaks that bond and releases IP3 into the cell, and diacylglycerol, being a fat, is going to stay with the membrane. Right. So this is that calcium metabolism thing I told you about earlier. Let's look at this figure though. This is one I made a while ago, and I think it's better than that. So here's norepinephrine activating alpha subunit. This should say GQ, not GP, which then activates phospholipase C, waka, 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 waka. And phospholipase C then breaks down PIP2 into IP3 and diacylglycerol. IP3 will come over to the calcium channel here. This is the IP3 receptor, and release calcium from inside the ER. Diacylglycerol will then activate protein kinase C, and protein kinase C will phosphorylate proteins. Some of them are going to be calcium channels, receptor operated calcium channels, and that will bring calcium in as well. So this whole system's all about bringing calcium up in the cell. And like I said earlier, calcium is so important for cells. I mean, it's a messenger that turns things on and turns things off. So it's tightly controlled. So this is how you can get calcium up in the cell. So this is classically how the smooth muscle contracts. If you remember back to when we talked about smooth muscle contraction, I said calcium increases in the cell. That's all I said. And from there, we went to calcium calmodulin and then activating myosin light chain kinase and unfolding the myosin molecules and all that. This is how it got up. So norepinephrine could be released onto an artery, activating an alpha receptor on the artery, activating IP3 release, calcium, and that causes calcium release, and then the muscle contracts, the smooth muscle contracts. Okay, so that's the IP3 calcium system. Now, like we did with the cyclic AMP system, let's look at how each step turns on, which steps are catalytic, which ones are stoichiometric, and which ones are amplifying, and how do we turn each step off? My brain just went somewhere else there for a second. Okay, so norepinephrine binding with the alpha receptor, stoichiometric, right? 1-to-1. How does it come off? Well, let's get back to that later. Okay, so alpha receptors get activated. They can activate multiple G proteins between 10 and 100, so that's catalytic. Okay, that's our first catalytic step. This GQ then activates phospholipase C. Phospholipase C and GQ binding to each other is stoichiometric, 1-to-1. Phospholipase C then goes off and breaks down every PIP2 it can find. So that's catalytic. It's going to break a bunch of those down. IP3 gets released into the cell, the cytoplasm. IP3 then comes over and binds to its IP3 receptor, stoichiometric, okay? I believe it's 1 IP3 to 1 receptor. It might be 4, I'm not really sure. Not important right now, but IP3 binding with the IP3 receptor is going to be stoichiometric. Once that opens, calcium comes flooding out of the thing. Obviously, that's going to be an amplification, right? 1 molecule of IP3 for a boatload of calcium coming out. And then that calcium can activate kinases. We saw in smooth muscle the calcium calmodulin dependent protein kinase and also myosin light chain kinase. So that would be stoichiometric, and then the phosphorylations by those kinases would be catalytic. Up here, phospholipase C makes a whole bunch of diacylglycerol. Every time it makes one of these, it makes one of these. This then binds to protein kinase C, stoichiometric. Protein kinase C goes off and phosphorylates every protein that has got the right consensus sequence. So we saw the consensus sequence for protein kinase A. This will have a different consensus sequence. So it's not phosphorylating the same sites, different sites on either the same or different proteins. And that is catalytic. And then these will let calcium in, which is then catalytic again. Okay. Most important catalytic ones to remember are the upper ones. Once we get down here, it's not as important. Okay, so that's how everything gets turned on. How do things get turned off? Again, your hormone binding receptor is not covalent. It jumps on and jumps off. When norepinephrine goes away outside the cell, it jumps off and it doesn't go back on. And so that turns that off. The alpha subunit activates the G protein. I don't show it here, but the G protein shuts itself off all the time, right? So we've got G protein cycling going on here. So that GQ then will break down GTP into GDP and shut itself off. Another thing that's not shown here is the whole beta gamma aspect, because it's just too much complication to put into this little figure. But that's going on there too. Okay, phospholipase C, when when GQ shuts off, that shuts phospholipase C off because it comes off the phospholipase C. So it stops breaking down PIP2, but we still have a whole bunch of IP3 in the cell. IP3 then gets broken down by phosphatases. So all those phosphates get taken off by phosphatases. Now you've just got free inositol laying around, and that gets recycled back into making PI again. Diacylglycerol will eventually, how does that get, I'm not sure how that gets out of there. There may be an enzyme that breaks it down, or it may just get recycled back into PI. I'm not really sure. But it will decrease eventually in the cell. Here's an interesting factoid. So this can be made into 2-AG, 2-arachidonyl glycerol, which is an agonist for the THC receptor. Yeah, the CB1 receptor. So that's your endogenous THC, diacylglycerol. One of that acyl group on the number 1 position is taken off, and what's left is your agonist for your CB1 receptor. Not important for this class. That's important for the drugs class. Okay, and then PKC gets turned off when this goes away, and then you've got phosphorylations here. You know how they get taken off, right? Phosphatases are taking them off. But now we still have calcium high in the cell. How do we get calcium out of the cytoplasm? Saw that in the last exam, right? Circa over here, and you also have plasma membrane calcium ATPases, and depending on the cell, you could have sodium calcium exchanger as well. Okay, and that's going to bring calcium back down in the cell. Wow, talking a lot today. So here's our calmodulin again, like we saw before. Calmodulin's got binding sites for 4 different calcium ions. When calcium binds to it, it changes conformation, and then it binds to some target protein and turns it on. We talked previously about turning on myosin light chain kinase. There's also a calcium calmodulin dependent kinase that will get turned on by it. And in the next class, we'll talk about activating another enzyme, nitric oxide synthase, with this calcium calmodulin. So calmodulin is kind of like a modulatory protein that uses calcium, calmodulin. Get it? So it's there in the cell, and it's when calcium increases, calcium binds to it, and then it activates other proteins. So it's kind of how calcium activates some proteins anyway. What does this system do in the cell? I'm glad you asked. Well, when thrombin binds to platelets, it causes them to stick together in order to make a clot in your blood. So if you have a damage in your system somewhere, it exposes proteins that aren't supposed to be exposed because you've got broken cells. That activates the platelets to release thrombin, which then activates the platelets to bind to each other. So autocrine stimulation of platelets forming a clot. Here is muscle contraction, smooth muscle. Acetylcholine will bind to smooth muscles in your GI tract, perhaps, and that will cause contraction of those muscles, again, through that mechanism we already talked about, and that's parasympathetic. Insulin secretion, again, parasympathetic in your pancreas. Amylase secretion in your mouth or your pancreas. I guess, yeah, just your pancreas there. And antidiuretic hormone in the liver actually causes like glycogen degradation. Not that important. None of this is really important. It's just, here's some examples of what it can do. I wouldn't ask you about this on an exam, so don't worry about these tables. I'm just kind of throwing these things out there to make it seem a little more concrete. I don't know about you guys, but I've had enough for one day. How was that? Good? Good? Okay, well, I'm going to let y'all go. You're all going already. And I hope to see you again on Thursday. We will have another quiz. Woo-hoo.